The Science Success Center, with funding from Title V, presents How Cells Harvest Chemical Energy, a biology workshop. Hi, I'm Steve. Life requires energy. A cell uses energy to build and maintain its structure, transport materials, manufacture products, move, grow, and reproduce. This energy ultimately comes from the sun. In cellular respiration, oxygen consumed as glucose is broken down to carbon dioxide and water, the cell captures the energy releasing ATP. Our starting components, glucose and oxygen, are consumed and used in the mitochondria, and our products are ATP, which is our chemical energy, and heat energy, and as well as carbon dioxide and water. Generating ATP for cellular work is the fundamental function of cellular respiration. The balanced chemical equation summarizes cell respiration as carried out by cells that use oxygen in harvesting energy from glucose. The equation tells us that atoms of the starting reactant molecules, glucose and oxygen, regroup to form the products carbon dioxide, water, and lastly ATP. Cellular respiration can produce up to 38 ATP molecules for each glucose molecule. The energy available to a cell is contained in the arrangement of electrons in the chemical bonds that hold an organic molecule like glucose. During cellular respiration, electrons are transferred to oxygen as the carbon and hydrogen bonds of glucose are broken and the hydrogen-oxygen bonds of water form. The movement of electrons from one molecule to another is an oxidation-reduction reaction, or redox reaction. In a redox reaction, the loss of electrons or hydrogen atoms from one substance is called oxidation. Notice here that glucose, which is made up of carbon, hydrogen, and oxygen, is converted into carbon dioxide. We say that this is an oxidation reaction because there is a loss of hydrogen, as we notice that carbon dioxide does not contain any more hydrogen atoms. Likewise, the addition of electrons or hydrogen atoms to another substance is called reduction. A molecule is said to become oxidized when it loses one or more electrons and reduced when it gains one or more electrons. Two key players in the process of this redox reaction are an enzyme called dehydrogenase, which is responsible for the oxidation part of this reaction because it removes hydrogen, and NAD+, which is an organic molecule that cells make to shuttle electrons in redox reactions, therefore is responsible for the reduction half of this reaction. Cellular respiration consists of a sequence of steps that can be divided into three main stages. Stage 1, glycolysis, occurs in the cytoplasmic fluid of the cell, that is, outside the organelles. Glycolysis begins respiration by breaking glucose into two molecules of a 3-carbon compound called pyruvate. Stage 2, the citric acid cycle, takes place within the mitochondria. It completes the breakdown of glucose by decomposing a derivative of pyruvate to carbon dioxide. The main function of glycolysis in the citric acid cycle is to supply the third stage of respiration with electrons. Stage 3, oxidative phosphorylation, involves the electron transport chain in a process known as chemoosmosis. NADH and related electron carrier FADH2 shuttle electrons to the electron transport chain embedded in the inner mitochondrial membrane. Most of the ATP produced by cellular respiration is generated by oxidative phosphorylation, which uses the energy released by the downhill fall of electrons from NADH and FADH2 to oxygen to phosphorylate ADP into ATP. Now let's begin discussing glycolysis, the first stage in cellular respiration. Glyco means sugar, while lysis means breakdown. Therefore, glycolysis is the breakdown of glucose, which is a sugar, into two pyruvate molecules. The compounds that form between the initial reactant glucose and the final product pyruvate are known as intermediates. The energy investment phase actually consumes energy. In this phase, ATP is used to energize a glucose molecule, which is then split into two small sugars that are now primed to release energy. Two ATPs are used in the energy investment phase. The energy payoff phase yields energy for the cell. In this phase, 2 NADH molecules are produced for each initial glucose molecule, and 4 ATP molecules are generated. Since the first phase uses 2 molecules of ATP, then the net gain to the cell is only 2 ATP molecules for each glucose molecule that enters glycolysis. These 2 ATP molecules from glycolysis accounts for only 5% of the energy that a cell can harvest from a glucose molecule. We will soon discuss the remaining 95% of energy that is produced. As pyruvate forms at the end of glycolysis, it is transported from the cytoplasm into a mitochondrion, the site of citric acid cycle. Pyruvate itself does not enter the citric acid cycle. A large multi-enzyme catalyzes 3 reactions. First, a carboxyl group is removed from the pyruvate and given off as the molecule of carbon dioxide. Secondly, the 2-carbon compound remaining is oxidized while a molecule of NAD is reduced to NADH. And thirdly, a compound called coenzyme A, derived from B vitamin, joins a 2-carbon group to form a molecule called acetylcoenzyme A. The citric acid cycle is often called the Krebs cycle in honor of Hans Krebs, the German-British researcher who worked out much of the cyclic phase of cellular respiration. First, coenzyme A helps the acetyl group enter the cycle and then splits off and is recycled. As the new acetyl group enters in the citric acid cycle, it is added to oxaloacetate to form citrate, a 6-carbon molecule. The citrate is then processed through a series of redox reactions. At step 2, the citrate is oxidized to form NADH and is stripped to form 2 carbon dioxide molecules. At step 3, ATP and NADH is generated. At step 4 and 5, FADH2, which is another electron carrier, and NADH are produced. Each turn of the cycle makes 1 ATP molecule by substrate level phosphorylation. It also produces 4 other energy-rich molecules, 3 NADH molecules and 1 molecule of the electron carrier, FADH2. We have now made it to our third and final step of cellular respiration. As mentioned before, in the citric acid cycle and glycolysis, there was a production of NADH and FADH2. These electron carriers carried electrons that were stripped from the glucose molecule and taken over here to the electron transport chain, which is located in the intermembrane space. The electron transport chain is made up of protein complexes and ATP synthesis. Now, what happens is that the NADH and FADH2 will leave their electrons, as shown right now, and they will pass through the protein complexes. As they pass through the protein complexes, they allow this hydrogen gradient to be produced within the intermembrane space. The electrons are then accepted by the oxygen molecule, and water is produced. Because the hydrogens cannot leak out of the mitochondria and into the cytoplasm, the hydrogens return back through the ATP synthase, and the exergonic energy that is released from this fall allows the production of ATP. Now, let us review cellular respiration. Glycolysis, occurring in the cytoplasmic fluid, and the citric acid cycle, occurring in the mitochondrial matrix, contribute a net total of 4 ATP per glucose molecule by substrate level phosphorylation. The cell harvests much more energy through oxidative phosphorylation, which yields an estimated 34 molecules of ATP. Therefore, the net yield of ATP molecules per glucose molecule has a theoretical maximum of about 38. Cellular respiration cannot undergo without the use of oxygen. However, fermentation provides an anaerobic path for recycling NADH back to NAD+. There are 2 types of fermentation: lactic acid fermentation and alcohol fermentation. In lactic acid fermentation, NADH is oxidized to NAD+, as pyruvate is reduced to lactate. The lactate that built up in muscle cells during strenuous exercise is carried in the blood to the liver, where it is converted back to pyruvate. In alcohol fermentation, yeasts are single-celled fungi that normally use aerobic respiration to process their food, but they also are able to survive in anaerobic environments. Yeast and certain bacteria recycle their NADH back to NAD+, while converting the pyruvate to carbon dioxide and ethanol. Thank you everyone for watching. Come visit us at the SSC if you have any questions. Good luck in all your studies and tune in for the next workshop.